All right. So, hello, everyone, and welcome to the Method Made podcast, where we discuss operational challenges in bioprocessing with a general focus in cell and gene therapy. Today, I'm joined with Moritz von Stosch. Welcome, Moritz. How are you?
Thank you. Thank you very much for having me. I'm doing very well. How about you?
I'm great. So, Moritz, to start with, I'd love to hear a little bit about your background in digital bioprocessing, process modeling, and cell and gene therapy.
With great pleasure. So ever since I finished my studies back at Aachen University in Germany, I went into the field of digital bioprocessing. I was very interested in hybrid modeling, which combines mechanistic or fundamental engineering knowledge with machine learning. And these tools were traditionally applied to bioprocess modeling. So this is how I went into bioprocess modeling. This is how I went into modeling as such. And then during my PhD, I had the opportunity to really apply it to all kinds of bioprocessing challenges, such as optimization, monitoring, control, so really the entire spectra. And because I was so interested in the technology as such, and I saw an opportunity, we afterwards, during my postdoc, also went into systems biology and explored also whether we could use it for synthetic biology, which was back at the time coming up. And we also started looking at founding a company, which was back in the time called HiPad, which didn't have much success back at the time. People were not so interested in digital bioprocessing. And then I got the opportunity to go to Newcastle University, where I got a lectureship position. And I was also there focused on improving process development, really bioprocess development in particular, really trying to get the number of experiments down that I needed in order to develop a process. And together with a colleague from GSK, I invented the method called intensified design of experiments, which allowed to do less experiments to develop the process. And shortly after, I joined GSK, where I was leading the Process Systems Biology and Engineering Center of Excellence. And also there, I was pretty much focused on using data, using models in order to accelerate bioprocess development, and of course, to make sure that the quality of the product would be on spec. And during my time at GSK, I was very much focused on microbial processes, even so there was also mammalian and mRNA activities ongoing in the drug substance department. And of course, there's not only upstream, there's also downstream. And then shortly afterwards, I saw an opportunity to join therapyHow. And therapyHow back at the time was focusing on hybrid modeling and in particular mammalian upstream processes, so the typical monoclonal antibody processes. And I joined as a chief innovation officer, really focused on extending the use of the solution to other fields, so cell and gene therapy, and then also mRNA. And on the other hand, I was also focused on finding partners with whom we can provide the technology, not only to the big pharma companies, but also to small biotech and cell and gene therapy companies, of course, small biotech. So this is a little bit in a nutshell, the development and how I came into the world of bioprocess modeling and digital bioprocessing.
Wow, what an extensive career so far. Well, I'm really excited to sort of discuss some of the area of cell and gene therapy that you worked in. And it'd also be great to hear sort of an overview of your work on sort of analytical challenges and scalability and standardization as well.
Oh yeah, with great pleasure. I mean, during the entire time in industry, so one thing when you come out of the academic track, right, you cannot imagine the challenges that are present in industry. And then you come to industry and you understand for the first time what the challenges really are. And analytics is one big challenge. Why is that? Just because across different projects, the analytics might vary. Across different companies, of course, the analytics vary. Across sites, the analytics might vary. You have different calibration standards and so forth. And then even in different instruments. And there might be that if you record one thing with a different instrument, there's a slight difference in the value. So, and then, of course, for many of the more new modalities, the analytics are not so well developed. That means analytical errors are high. It's not, if you repeat the same thing over and over, you might have variations of 10, 15, 20%, which also historically, for instance, you had with ELISA assays and so forth, which of course, with the improvements in technology, decreased over time. So yeah, this being said, analytics is really one big thing which I stumbled upon when I joined industry. And then standardization, of course, is another big thing, making sure that everyone repeats the same process over and over, and that the process is not only identical or similar, at least in the lab, but that the results can also be reproduced elsewhere, right? Not necessarily replicated, but at least reproduced, and calling things the same thing and recording the same things. That is, of course, a huge challenge, in particular in the data world. I mean, there's more and more standards coming up, but people, and that includes myself, I mean, standards are really important to have and important to adhere to, but people are not very good with that, and that includes myself. Yeah, and we can jump into the challenges that arise from that as well. And the last point you touched upon was scalability, and I remember that back in the time when I was at university, I took a course on bioprocessing, and Professor Bux told us that one of the main remaining challenges, and that was back in 2002, 2003, one of the main challenges in bioprocessing is scalability. And ever since, this challenge has accompanied my career. Scalability is still a big challenge, right? We're getting better and better in addressing it and understanding it. We have tools such as computational fluid dynamics and various versions and variations thereof to characterize the reactors better, to understand flows better, to understand really also heterogeneities, which we have in the reactor better in terms of material and both temperature and so force fields. But yeah, it remains a challenge. So gene and cell therapy, of course, is no exception there. On the contrary, perhaps.
That's amazing to hear. Well, that's sort of exactly the next thing I'd love to touch on is I'd love to hear sort of what specific challenges you see in cell and gene therapy process development. Maybe we could start with sort of analytics that you were just discussing. You mentioned ELISAs. I'd love to hear sort of, yeah, what the current challenges are. Where are we at with that?
Yes, I mean, from a modeling point of view, of course, analytics are very important. If we cannot measure it, or if we cannot measure it accurately, then we also have a hard time modeling it, right? And I think there's analytic challenges on both on the input side, and with input, I mean really all the things that might impact process performance, and also on the output, on the response side. So how much tighter are we getting? What is the product quality? So capsids, empty, full capsids, right? And that ratio. You mentioned ELISA, which of course, yeah, might or might not be very reproducible. There's other techniques, new techniques arising. People are looking at cells with microscopes, with cameras, trying to understand, are these cells transfected? Is there a change in the presence of the cell, and in the morphology, exactly. And yeah, I think we have, industry is getting better and better in converging towards standards, and certain standards are used also, of course, supported by what the regulators want to see. But we still see significant variations, in particular when measuring process response. We see still variation across different companies as well. And I'm not sure that all the time we also have available all the sources of variation. So for instance, variation in raw materials and so forth. While we might look at lot numbers, we don't really cover what is changing from lot to lot, right?
Okay, yeah, that makes a lot of sense. So we briefly talked about sort of capsid filling and sort of ratios of full capsids. I'd love to talk about these sort of specific challenges with transfection in gene therapy, how that might affect these sort of quality outcomes you were just mentioning.
Yeah, I mean, in particular, the AAV platform, for instance, we have the transfection step in this in this process, and the transfection, yeah, comes with a lot of challenges. I mean, one of the if we look at CHO, right, and monoclonal antibodies, one of the big steps was that we had this stable producer cell lines that no longer needed transfection. We had a cell line, and we just needed to integrate the monoclonal antibody sequence into the DNA, and there's different ways of doing that. And essentially that led to really the process platforms which we saw in CHO, and then the rise of the monoclonal antibody titers over the last decades. And I think for gene therapy, it could be very similar, right? We're looking still at transfection processes. These transfection processes have issues with the stability. There is scalability issues, and then the efficiency of transfection is also something that is many times investigated with several different parameters, and material might be screened to increase transfection stability. And then, of course, this also has an impact on how many capsids we get, and perhaps also the full to empty capsid ratio.
And I'm curious, with these these kind of ratios, does that variation right in today's sort of state of the art, does that reach all the way to sort of the end patient, or are there currently sort of quality methods for controlling that?
Yeah, I mean, there's many methods. Well, this process is upstream, right? So we still have all the way through the purification to improve the full to empty, the full to empty ratio. And typically in purification, you would try to increase, of course, the full the amount of full capsids. And yeah, one thing is, of course, to having the methods in place to increase the amount of full capsids. The other thing is measuring and being able to evaluate that the method actually works well. There's different ways of doing that. Digital PCR or and so forth being one of which. So yeah, there is of course ways to do that and to consistently get a certain amount of full to empty. But if you could get it right first time, right, if you could get it upstream, then you would have much more leverage on the downstream operation to only sort out few empty capsids. Okay,
so currently the sort of control step is downstream to sort of figure out the amount of full capsids, but sounds like down the line the goal is to have the right amount of full capsids starting at upstream. Makes a lot of sense.
Absolutely.
So the next thing I'd love to talk about is data quality and quantity. So when you're building these models, like what kind of problems do you encounter with the underlying process data? Like I see a lot of these new modeling companies that are delivering platforms, and some of them, it looks like maybe assuming a high quality level of the underlying data. And so, yeah, I would love to hear sort of the reality of that current situation.
I think data quality and quantity is really something that is it lays the foundation for the quality of the model, right? And I think one thing which is important to understand is that if you want to have a certain insight into the process and you want to gain that insight through modeling, then you also need to have the data to go with that. For instance, if you want to understand how the process evolves over time, you need measurements over time. Otherwise, there's no way to understand how the process will evolve. And we see, for instance, that if we have shake flask experiments, or that we have experiments in bioreactors in very early stages where only endpoint samples are taken, that might be fine for material screening, but that does not give us an indication of how does the process really evolve over time. And so sampling more could help to reuse that data later stages, because we essentially understand how the process evolves in early stage, and then in later stage we can compare the evolution of the early and the late stage process. And the same is true for shake flasks. And then, I mean, shake flasks, they come with their own challenges. Of course, in shake flasks, you can very easily parallelize them, right? This is nice for data quantity. But then on the other hand, you can, for instance, not necessarily control pH or the soft oxygen. The conditions are not so well controlled, and that might give rise to variation that you might not be even capturing, right? Because many times pH is not systematically measured. And that, of course, means that, data quantity is there, but data quality decreases. So that is one of the challenges that arises. And then I mentioned material attributes. I think earlier, if you have variations in raw material, if you have variations in lots, or if you have variations in the n-1, so in the bioreactor or in the shake flask that you use in order to actuate your production process, right? That also leads to variations, and we have seen that many times. The expansion of cells might impact on the production stage, in particular on the transfection, and so yeah, all these steps need to be considered and considered also in the analytic strategy.
Okay, so I would love to sort of dig a bit deeper into, like you mentioned, time period based measurements as opposed to just endpoint measurements. Where is that, like, where is that most important through the whole bioprocess?
Well, I think if you repeat the same process over and over again, it might be nice to have 2 or 3 series of time data, of 2 or 3 series of data over time, but you don't need to do it all the time, in particular if you have the process well under control. On the other hand, if you then perform manipulations, say for instance the temperature profile, or you start feeding, or you start the transfection process, and you might as well vary the time where you do that, then of course it would be nice to have a measurement before so that you know where is the starting point, and then measurements afterwards so that you know how the process responds to that change that you introduce into the process. And in particular, if you have multiple changes, or if you have not a discrete change, but a continuous change, for instance, a feeding ramp or so forth, then having multiple measurements over the time it significantly increases the understanding. So that certainly helps.
Okay, that makes sense. It's sort of generally the you need dynamic data for dynamic modeling.
And absolutely true. Absolutely true.
Well, that's great to understand. So I'd love to hear a bit about the standardization gaps. So, like, how do raw material variations and these process inconsistencies impact the modeling accuracy?
Yeah, I mean, standardization gaps, you mentioned it, raw materials, and in particular, we record raw materials mostly at the moment with lot variations, right? There's not a lot we record on the material itself. We just say this is lot 1, this is lot 2, lot 3. And so this is providing a challenge, because essentially, if you don't have the same kind of material across sites, or if you don't have the same kind of material even at the site, and you might have variations in lot, you don't understand how a new lot will impact on your process unless you have run 1 or 2 runs with this new lot, right? Then that is certainly one challenge. Then there's of course different naming challenges which you might have if you have different sites in different countries. Yeah, we were well, at GSK we had a site in Italy and a site in France, right? And if you look at the abbreviations, which of course follow the way variables or namings are used in France and in Italy, then in French we saw the inverse in words. So we had dissolved oxygen being OD rather than DO, because it's oxygen dissolvé. And yeah, so that comes with challenges, which of course need to be carefully handled. And yeah, whenever you're also changing sites, you're changing from, for instance, process development to pilot plant, if the site is big enough, right? Then of course all these things which are assumed, hopefully written down, but many times also assumed, right? They may not be properly tech transferred to the next site, and there's of course room for interpretation, and whenever there's room for interpretation, there's room for variations in the process as well. And I think there's this fair data principle. So there's of course Alcoa Plus, right, or Alcoa, which is coming from the regulatory body, making sure that the data is captured and done in such a way that in principle the experiment could be reproduced. But the FAIR principle goes one step further, in that the data should not only allow to reproduce the process, but also should be usable in the future, and you could essentially gain additional insight from the data in the future. And when we look into that, then standardization is very, very important. And that is why I think so many initiatives, such as the NAMU initiative, or the allotrope initiative, or yeah, there's many others. There's the CELA, for instance. Yeah, why they are there and why they have their right of existence, right?
Yeah, I've heard quite a bit about this FAIR principle, and I also see that there's some sort of new initiatives that are. I'm hearing that a lot of it is about documentation as well, like documenting everything that's required for processes. And you mentioned sort of scaling as one of these challenges. So I'd love to go a bit deeper into this scaling challenge. You know, we've talked about shake flasks. I love to kind of walk through sort of from shake flasks through to industrial scale manufacturing, how these gaps sort of compound when companies try to scale processes. I've heard when I talk to process development engineers, this is one of the biggest and most common things that comes up.
No, absolutely true. I think scalability is a big issue, and many companies seek to use modeling in order to improve scalability. What we see is that a lot of screening is performed at the shake flask, not giving much thought to how the process will scale to the larger scale, which is fine, right? You're just screening, you're just ranking. And then the first process development activities, where you really look at process parameters and so forth, they are typically performed in bioreactors, typically small scale, high throughput, because of course you still want to characterize your process very well at this stage. And then there is some first runs in larger scales, typically also linked to the production of material for clinical phase 1.
Mhm.
And hopefully, and many times that works, at the larger scale, there is a process, or the process that was developed at the smaller scale still performs well enough to produce the material. And then, of course, the data from that larger scale can be used together with the small scale data to develop a physical scale down model, right? And this physical scale down model in principle should emulate how the process behaves at the larger scale, but emulate that at the small scale. And then going forward, phase 2, phase 3 activities where we come into process characterization and into optimization, we can use that scale down model to really evaluate how process parameters and material changes impact on process performance. But the development of the scale down model, of the physical scale down model, and scale up as such is still a challenge. So I was mentioning before that there is computational fluid dynamics, for instance, being deployed in order to characterize the fields, the material flows, the transport phenomena in the bioreactors better, therefore get also better insight. There's ways of making that also fairly simple by breaking down the bioreactor into certain regions that have a more or less consistent flow pattern, and then characterizing this by typical engineering numbers such as Reynolds or tip speed, or the typical numbers, and using those perhaps in the scale-up strategy. But I think what is clear in the bioprocess industry is that for upstream there is no linear scale-up, right? Upstream is nonlinear. The parameters that we have, they interact with each other, so we cannot just simply linearly scale up these parameters. And we humans, we are not very good when it comes to interaction of parameters and understanding the impact on the process, whereas models, this is exactly where these models accelerate, right? This is exactly where machine learning is very good. So in principle, if we have data and if we did the characterization work well, then we should be able to scale up.
Okay, well, yeah, so we discussed a little bit about engineering numbers like Reynolds number, and one of the things that has come up in a few of these conversations that seems to be a challenge with scaling is homogeneity and mixing. So my understanding is that with shake flasks, sort of dissolution mixing happens very effectively, and that with larger scale reactors, you get these sort of pockets that may lack oxygen, and there's some sort of heterogeneity. Are the current systems like digital twins very good at modeling these types of things?
Well, yes, I think there is modeling solutions to characterize these zones very well, and you can use then these solutions also to drive reactor design, right? Trying to improve the overall homogeneity of the material that is present in the reactor. And I think that might be very important as well in order to get similar performance across the scale. The other way that one can use these simulations is to construct physical reactors on the small scale, where you have, for instance, two reactors sitting next to each other and flow is exchanged, or there's a plug flow reactor next to a steer tank reactor, to emulate that there's these gradients at the larger scale, and that therefore, if you cannot get rid of the heterogeneity at the larger scale, you can emulate the heterogeneity at the smaller scale. So this would be, for instance, one solution to look at that. Again, I think if you can also emulate this heterogeneities using operational parameters, for instance, steering rate or gas flow, which might not be entirely the same thing as if you build a physical second reactor, but at least it will give you an understanding of how heterogeneities drive your performance. And so if you characterize this well, if you have the data and if you use that in a model, likely you can use that to also gain insights on how changes in these parameters might impact on the performance at the larger scale.
Yeah, that makes a lot of sense. And I'm starting to see some emerging companies that one of the products are like inserts for small scale bioreactors that aim to sort of mimic these differences. The next thing I'd like to talk about is communication and documentation practices. So we've talked a little bit about data quality. So how do poor communication documentation practices affect the quality of data going into these models?
Yeah, I mean, documentation practices is one thing you can really get me going on.
Okay.
So if we didn't document it, if we didn't measure it, then we'll have a hard time with the modeling or making the model understand that something changes, right? The model doesn't see the change, so it cannot predict that there will be a change in the process if it didn't see the source of variation. And that, of course, means that, hopefully first we understand what is driving variation in the process, and we document and measure this. And second, when we, develop the process, we have enough experiments where we try to reproduce, we try to replicate the results to understand how much batch-to-batch variation do we have. So that is certainly one thing that is very important. Another thing that we also see is that we should not only record the values that should be. So for instance, pH should be at 5, but in fact, you want to record the value that is really present in the reactor. So you might want to measure pH, and okay, measuring pH might also have to do with the calibration of the pH measurement and so forth. But yeah, just to give you that that understanding, and then you might take a sample, you might label the sample wrongly, and therefore it is not entirely clear when the sample was taken, and all these kind of things might impact really on the quality of the model afterwards. And then, as I mentioned before, the batch-to-batch variation might also come from variations in the raw material that we have not well characterized. So there's plenty of sources for perhaps modeling problems when documentation and the data is not well recorded, is not well captured.
Yeah, well, yeah, you mentioned that example with the pH, and something that I've sort of noticed is that depending on the circumstances, sometimes these documentation can be more aspirational than reflective of real measurements, and that is always a source of error. And yeah, it makes a lot of sense.
Yeah, I mean, on the other hand, of course, the model can also help you to understand whether or not the degree to which you document and you capture the process is sufficient to reproduce it, right? Because if the model cannot reproduce the behavior, and supposedly the model can reproduce everything, right? Because, for instance, machine learning model, as long as there is sufficient data, whatever that means, we should be able to capture the process behavior, right? If we cannot do this based on the data and documentation that we have, we might ask ourselves, do we document, do we capture enough data to allow ourselves to really understand what is going on. And yeah, the model might help us directly in this sense, right?
Okay, that makes a lot of sense. Last thing on this documentation and recording that I'd like to touch on is the disconnect between recording and observation. So how does the separation of tasks between the operators who actually run the process and the analysts that are doing the modeling impact the data quality? Is it, yeah, I'd love to hear a bit more about that.
Yeah, you're absolutely right. I mean, depending on how the processes are developed, the samples might not even be analyzed by the same people who take the sample, right? And so there's certainly separations of tasks. There's the separation. There might be a separation between the person running the process, the person analyzing the samples, the person putting that information together and analyzing how the experiment evolved, and then another person who might do the modeling. So there's a lot of people involved. And many times the people who, for instance, run the process, they're interested in different quantities or different numbers than the person who analyzes the process and the process data, and then the person who models the process, right? And because the persons might be interested in different data, they also look at different quality metrics of the data differently, which means that there might be certain outliers that are introduced in the data by accident, by typing up the wrong number, by a copy and paste error, and so forth. And essentially, this data are never captured by the people who are recording the data. Why? Because for the people who are recording the data, this recording is not the major outcome. They do it because they're asked to do it, and yeah, there's a way for it. And of course, they're not purposefully introducing errors, right? These errors they slip in by chance, but because there's no way of directly reviewing the data necessarily, there's no win for these people to review the data, right? They're not doing something with the data afterwards. They might not capture it. And so these errors are captured at a later stage. There might not be ways to directly correct for them. So these data errors go away. There might be information missing how the process is run, and just assumptions. The modeler might make assumptions of how the process is run. For instance, when the process is fed, when its feeding started, when the process might be transfected, when its transfection started, how the profile might look of transfection and so forth. And so all these things all these uncertainties that stem from the separation of the task, right, that might cause variation in the process. And we also see it when we go from R&D scale or process development scale to pilot scale and then to manufacturing. We have the same kind of separation between groups. It might be different people, and of course there's also operational or organizational ways of dealing with that. But if there's different people there might be assumptions that are taken, and therefore variations that might be introduced to the process. And the same is true for the modeling, as I mentioned.
Yeah, well, what I'm hearing is that a lot of these assumptions, they can really carry a lot of risk, because it sounds like at the end, the model may get data that carries these assumptions through, that are, if they're not accounted for, are going to impact the end result. Is that right?
Well, you could formulate it this way. I see the risk is, I think the risk is not that high. Why? Because typically we don't develop models for the purpose of modeling. We develop models to do something with the models, or at least hopefully. I mean, if you use the model to design the process, and you then, for instance, measure for that newly run experiment the process response, and it's completely off, you might ask yourself, of course, why is the process response completely off? So typically you find that assumptions were not correct and so forth. So that there is this correction step typically in all process development activities. The process development activities are anyways iterative, so you will capture those. But do we need those in the first place, right? Do we need to capture those errors? So I think by systematically documenting and systematically using models in particular, we might get much faster and much better in the recording of the data, the use of the information, and then speed and efficiency of process development.
Okay. So something else I'm quite curious about along these lines of sort of multiple operators involved in these things, I'd love to discuss knowledge transfer a bit and sort of how this plays into this. Is maybe we could talk about the knowledge transfer, say, between the operators who actually run the experiments and then the modelers and people analyzing them.
Yeah, I mean, knowledge transfer is ultra important, right? And it's perhaps, yeah. I'm not sure whether it's always called knowledge transfer or knowledge management, but I think it's the essence of process development and of tech transfer. And I think there is several different ways to improve knowledge transfer and to use, in particular, also models to transfer the knowledge. The first way, I think, is that rather than separating the tasks by people, one person to develop the process, one person to analyze it, and so forth, we can integrate much of that work using, for instance, a digital platform. And if we do that, if we really democratize access, for instance, to process modeling by simplifying the way we develop process models, the person that records the process and that is that is responsible for running the process, that same person can model the process. And if the person does that, errors in the data might become much more quickly obvious, and the person who run the process anyways knows much better how to correct for those errors than the person who would have otherwise separately modeled the process. So this is one way of doing it, really democratizing access to technology. Mm-hmm. And I think the other way of doing it is using models as a knowledge transfer vehicle along with the data. What I mean is that if you develop the process, you at the same time that you develop the process, develop the model. So it's a simultaneous development of the two, and your understanding of the process is essentially captured in the model. And that means if a new person comes aboard, that person might read the documentation, but that person can also interrogate the model. It can, for instance, ask, what if I change temperature at that particular setting? And based on the data and evidence that has been gathered in the past, the model will simulate how the process will behave. And along with that simulation, there will also be a measure of uncertainty. How sure is the model that the behavior will really be such that it's simulated? And so I think the model really can become the knowledge transfer vehicle. It can help to transfer knowledge from the smaller scale to the larger scale. It can help to transfer knowledge across different molecules, across different loadings, for instance, payloads. And it can also help to transfer knowledge between different people, different operators.
Yeah, that sounds like, I think that would be a great direction. And it makes a lot of sense given the sort of assumptions between operators that by democratizing access to the technology, you can sort of reduce the work a single person has to do such that both the designer, they can both design and execute the experiments. And I guess that will reduce many of these interpersonal assumptions that might be sort of operational inefficiencies.
Yes, and that would be exactly my hope as well. Exactly my hope.
Okay, so I'd love to sort of, now that we're getting towards the end, talk a bit about the future, where you see this going. So I'd love to hear, in terms of generative AI, where you see the potential is to change some of the challenges we talked about.
Yeah, I mean, in particular, when it comes to documentation, right, and writing documentation and also gathering data, I think Gen AI has the capability to help making data recording much more systematic. It might help to extract even data and information from past documents and to represent them in tables and or data structures that we can use with the modeling. So not only qualitative information, but really turning it into quantitative information that can be used for modeling purposes. So I think in this way, it's certainly helping. And then I think going forward, the other big step that Gen AI could help with is the collaboration with the machine. If we can express ourselves much more naturally and we can say what we want rather than clicking through menus and assuming stuff, right, we might have a much better outcome of the exercise. And again, it goes also into the area of democratizing access, really giving people the chance to do process models that until today have no chance of doing that because there's certain expertise required for machine learning, for mechanistic modeling that is not available to that people.
Yeah, well, so you mentioned sort of providing access to interacting with machines to people that previously couldn't. And that's what I'm seeing one of the big innovations with natural language processing and the sort of commoditization of language models is exactly this, like on encoding and that type of thing. It seems that's one of the big wins happening. I'd love to hear, since we're sort of focused on cell and gene therapy, what sort of recommendations you might have for cell and gene companies that they might prioritize to improve data quality and quantity, documentation, and scalability?
Yeah, well, that's a big thing. I mean, what can we recommend? I think very important is that these companies don't have a process development strategy in the traditional way, but that these companies adopt also a development strategy where models, and that could be machine learning models or mechanistic models or a combination thereof, hybrid models, are used to develop the process. I think if these models are used, it helps to consistently capture the data that is needed. And there, of course, that should be done in a systematic way and hopefully in such a way that the people who record the data also benefit from the quality of the data because they model it. So I think process modeling really can help these companies to improve data consistency, data quality, to improve the process understanding, and then to also translate that process understanding and to transfer it across different scales. So my recommendation would really be to go into modeling and to using modeling for process development work. And just to be entirely clear, we're not modeling for the purpose of modeling. We're modeling for the purpose of developing the process, right? Of answering process development questions. And so adopting models for that purpose, I think, would be my big recommendation. And best not only doing that with a modeling expert, but really democratizing access and have different people modeling the process and only have the expert for those cases where it's really hard and a process scientist might not be able to derive a model that is really mimicking process behavior.
Okay, that makes a lot of sense that you really need to have sort of the goal in mind of the process outcomes you're looking for when using and even designing these models so that, yeah, as you said, you don't end up in a situation of modeling for its own sake.
Absolutely, absolutely. And I mean, I think something which we also see is that model accuracy is many times taken too serious, right? If you have a model which is good enough to answer the question at hand, that model is good enough to answer the question at hand. If you then invest 1, 2, 3, 4 months to improve model quality by just a little bit, I think this additional effort is not warranted because essentially running another experiment would have increased model accuracy beyond what you can achieve when investing 3 or 4 months into the modeling, and you would have gotten the results much faster with much less effort. So I think that is also something that needs to be carefully kept in mind when using the model for process development purposes.
Well, this is something that's actually come up quite a bit, is the sort of risk of over-optimization. And one perspective that I sort of appreciate in this area is that in this area of process development, good enough is perfect. And once you sort of have your standards in place and you've reached them, there is no real value in continuing optimization past that.
Fully agree, fully agree. Knowing when to stop is one of the most important things. And we many times called it phase-appropriate efforts or phase-appropriate process development. Yeah, I have good memories of that from my time at GSK.
Wow. Well, it's been a super interesting conversation today, Moritz. Thank you so much for joining us on today's episode.
Thanks so much for having me.